I'd like to welcome you today. Thank you for joining us. My name is Amy Layton. I am the Work-Life Program Coordinator for Cornell. I'm joined today by my colleague Diane Braddock, who is our Work-Life Consultant. And we are super excited to welcome the Schaffer Nishimura lab today. Sorry, Nozomi, who will share with us some of their groundbreaking research on Alzheimer's. We are very fortunate to have such a wonderful lab on campus. Before we get started, I just wanted to remind you of some upcoming events that we have. We have flyers up front here. We have a new cancer support group on campus. We have a Finger Lakes Independence Center demo day next week. They have all kinds of assistive devices for people that have surgery or have any kind of disability. So they're going to come and kind of do a show and tell for us. And there's also information on the Senior Living Expo for 2019. Bathrooms are straight down the hall towards large windows on the right-hand side. And with that, I'm going to turn it over to Nozomi, Chris, and Pietro. Thanks, guys.
Great. Good afternoon, everyone. So my name is Chris Schaffer. I'm a faculty member here in the Biomedical Engineering Department, as is Nozomi Nishimura. She and I are partners, and we jointly run our lab together. We also have Pietro with us today. Pietro runs the Human Computation Institute, which has worked with us for some of the data analysis for the project I'm going to tell you about. And you'll hear about some exciting opportunities to become intimately involved with our data at the end of this. So today we're going to talk to you about some work that's been going on in my and Nozomi's lab for about the last 10 years, where we're trying to understand a specific symptom of Alzheimer's disease, namely that patients have reduced brain blood flow. And we're trying to understand what causes that and what the consequence of it is. Now, before we get too far, we have some really exciting findings to share, but I want to tell you, they are all in mice so far, okay? So this is all in mice. Okay. So broadly speaking, my and Nozomi's lab focuses on, just one second, sorry. Broadly speaking, my and Nozomi's lab focuses on trying to understand how the interaction of different kinds of cells in the brain give rise to specific symptoms of neurological diseases. So how cell A interacts with cell B to give rise to a specific symptom of a neurological disease. And it turns out that trying to look at things at that kind of spatial scale, where you want to see individual cells and how they interact with each other, it turns out light is a good way to do that. Microscopy kind of techniques are a good way to do that. So our lab, Nozomi and I are both trained in physics, and actually a good fraction of what we do is developing those kinds of optical tools to be able to directly look at how different kinds of cells interact with each other in the brains of mice to give rise to specific symptoms of neurological diseases. So the disease we're going to tell you about today is Alzheimer's disease. I'm sure if we quickly did a show of hands, how many people have experienced Alzheimer's disease as a close friend or their family? So that is 100% of the room. And it's really a terrifying thing. While this disease has been described for over 100 years, the first paper by Dr. Alzheimer himself was in 1907. It actually is poised to become the pandemic of the 21st century. And the sort of the burden of this disease, both for people who have it, for their family members, and for society as a whole, is huge. So 1 in 8 Americans over 65 have Alzheimer's disease. 1 in 2 over 85 have Alzheimer's disease. This is a disease that really strips away your sense of self. It's characterized by progressive loss of the ability to form memories. So one of the earliest symptoms you'll notice in people developing Alzheimer's disease is you'll be having a conversation with them, and it'll feel pretty normal. But then they'll ask a question that they just asked like 2 or 3 minutes ago. And the reason is that the previous time when you asked that question, they were able to answer it, but the part of their brain that converts that experience into a memory is broken. And so they're not able to remember that they just asked the question or the answer that you gave. Over time, there's actually a loss of pre-existing memories and eventually loss of other neurological functions. To give you a sense of how bad it is, this is a picture of Auguste Dieter. She was the first patient who was diagnosed as having Alzheimer's disease. In addition to kind of looking dejected in this photo, around the time this photo was taken, she was asked to write her name on this small piece of paper that's pictured here in the lower left. And if you look, it took her 3 tries, but she eventually got her first name, Auguste. She was not able to remember her last name, like that was gone. In addition to this disease directly affecting the people that have it, it's a disease that also has devastating impacts on caregivers. There's millions and millions of hours of unpaid medical care given to Alzheimer patients by friends and family. And those caregivers in turn show signs of physiological stress and disease, as well as neurological disease. In addition, this is a very expensive disease. So in 2012, there was about $200 billion spent in the United States on direct medical care for Alzheimer's patients. This is projected to grow to be the entirety of the Medicare budget by 2050. Currently, there are no disease-modifying treatments for Alzheimer's disease. There's no drug or therapy or surgery or anything like that that you can do that will stop the progression or even significantly ameliorate the symptoms of Alzheimer's disease. So it's a pretty grim situation right now. I want to just very quickly give you a kind of a high-level overview about what's known about what causes Alzheimer's disease. So inside your neurons, in the membrane of those cells, there's a protein called amyloid precursor protein that kind of sits in the membrane. This protein is important for the kind of molecular level decision as to whether you should keep or eliminate a connection between 2 neurons, a synaptic connection between neurons, when you're learning things. So it's a very important protein in terms of how your brain is wired together. And when it's doing that job, it gets cut by a couple of enzymes that sit in the membrane and secretes this small protein or small peptide called amyloid beta. And that gets secreted into the space outside of cells in the brain. Now, this is happening in everybody's brain all the time. You're constantly making this amyloid beta that secretes into the extracellular space. By itself, single molecules of amyloid beta do not appear to be pathogenic in any way. But it turns out that amyloid beta is very self-sticky. So if 2 molecules of amyloid beta come together, they tend to stick. And then more and more and more of them stick. And eventually you get these aggregates of many tens to hundreds to thousands of molecules of amyloid beta all stuck together. And this makes this big kind of sticky protein mass that just, it's sort of like pouring glue in the engine. It just sticks to all kinds of different cell surface receptors and other things in the brain, and it messes things up. It leads to death of neurons. It impairs the ability of neurons to transmit information from one neuron to another. It drives increased inflammation in the brain and causes widespread problems. Eventually, this is a histological cross-section from the brain of a human that died with Alzheimer's disease. The little purple dots are the nuclei of cells, and these big sort of purple, brown aggregates, those are actually just lots and lots and lots of this amyloid beta protein all deposited and aggregated together. And probably when talking about Alzheimer's disease, you've heard the phrase amyloid plaques or senile plaques or something like that. So that's what people are talking about here, these amyloid plaques in the brain. So when neurons die, when they can't transmit information properly, when these things happen, the thought is that's what's impairing the cognitive function. In addition, at advanced stages of the Alzheimer's disease, as all of these neurons die, you actually just get a huge shrinkage of the brain. Like the brain physically gets smaller because the cells and things like that make it up have gone away. So then you might ask, well, what causes Alzheimer's disease then? What makes it more likely that you're going to have these amyloid beta aggregates that cause problems with neurons and other cells? And it is relatively simple. Basically, the higher the concentration of amyloid beta in the brain, the more likely 2 molecules of amyloid beta coming together is. Because if there's more of them around, there's more of them they could bump into. And so the more likely you are to form aggregates. And it's the aggregates that drive the disease. So there's basically 2 ways you could get a higher concentration of amyloid beta in the brain. Either you could produce too much amyloid beta, or you could fail to clear it from the brain. Because normally that amyloid beta that's produced by neurons it gets cleared out of it either gets metabolized, gets broken down in the brain, it gets cleared out of the brain through the blood, or some of it gets cleared out of the brain through space along the outside of blood vessels and drains into the lymph nodes. So things that either increase the concentration of amyloid beta in the brain or decrease the clearance of amyloid beta from the brain will tend to cause Alzheimer's disease. Now, there's kind of two broad classes of Alzheimer's disease. About 0.5% of cases around the world of Alzheimer's are what's called familial or early onset Alzheimer's disease. These are all a kind of a genetic form of the disease where there's genetic mutations either in that amyloid precursor protein or in those enzymes that cut it. And those mutations all tend to lead to increased production of amyloid beta in the brain. They all bias toward making more amyloid beta. So these folks, they tend to get the disease early on. Depending on the kinds of mutations, some of this is like mid-40s, early 50s. Some is more like late 50s, early 60s. Most people with Alzheimer's disease, like 99.5% of cases, have what's called the sporadic form of the disease, where there's not a specific genetic mutation that guarantees you're going to get it. For those patients, the disease looks similar, but it starts later and it progresses more slowly. There are genetic risk factors. So there's some gene variants that if you have them, it increases the likelihood that you'll get Alzheimer's disease, but it's not determinative. It doesn't guarantee you will get it. There's a lot of modifiable risk factors that are associated with increased incidence and severity of the disease. Many of them are kind of cardiovascular in nature. So things like having high blood pressure or having high cholesterol or diabetes or insulin resistance, things like that, all increase the likelihood and severity of Alzheimer's disease. In addition, head trauma increases the likelihood and severity of Alzheimer's disease. Every single concussion you've had in your life produces a measurable increase in risk for Alzheimer's. And there's some things that are protective. So educational attainment, believe it or not, looks to be protective against Alzheimer's disease. Regular exercise is clearly protective against Alzheimer's disease. Okay, so two broad classes of the disease. Only about 0.5% of people have this familial form, but it turns out for the research that we do on Alzheimer's disease, this form of the disease is really essential. And the reason for that is a lot of the studies we want to do, we can't do in people unless anybody wants to volunteer for us to drill a hole in their head and put a microscope so we can look and see. The things that we do are quite invasive. And so we're not going to do the research in people. Instead, we're going to do the research in mice. But mice don't get Alzheimer's disease. They don't naturally get it. So in order to try to reproduce Alzheimer's disease in mice, what researchers have done is taken the genes that are associated with these early onset forms of the disease and insert them into the genome of mice. So these days, there's incredible power to manipulate the DNA of mice and many other species. And for actually a couple of decades now, there's been mice that have been engineered in this way, where they have the mutated human DNA that leads to these familial forms of Alzheimer's disease. So these animals get the amyloid plaques, just like you find in humans. They get neural loss. They have learning memory problems, like when you're having a conversation with them, they'll keep asking you the same question. Okay, but these genetic models, they give us the tool to be able to study the mechanisms of disease using more invasive processes so that we could identify potential therapeutic strategies. Okay, so that's my kind of overview of Alzheimer's disease and the mouse models that we use to study it. Now what I want to do is talk about the specific symptom that we're interested in trying to understand. We're trying to understand which cells interacting with which other cells causes this symptom. So it turns out that patients who have Alzheimer's disease have a pretty big reduction in blood flow to their brain. It's about a 30% reduction in blood flow, and that's a lot. For perspective, you know when you're laying down and you stand up too quick and you kind of feel dizzy for a second? That's about a 30% reduction in blood flow. In that case, just for a couple of seconds before blood vessels in your neck dilate to increase flow to the brain. So Alzheimer patients are walking around with that level of blood flow reduction basically all the time. And so it's likely that that blood flow reduction contributes to the memory problems and other cognitive symptoms of the disease. People have known about this blood flow reduction for decades, but the mechanism has been unclear. And if you don't know what causes the blood flow reduction, it's really hard to figure out how to treat it. These mouse models, these genetically engineered mice that get Alzheimer's disease, they also show this same blood flow reduction. So that's an indication that the mice are good at reproducing at least this aspect of the disease that we see in humans. Another important thing is that this is an early feature of the disease. So if the blood flow reduction, like if that was something that happened right at the end stages of Alzheimer's disease, to be frank, I would be a little bit less excited about chasing it, because even if we fixed it, how much help are we likely to provide? But it turns out that this blood flow reduction, it's one of the earliest things that you see in patients that are developing Alzheimer's disease. It's one of the earliest physiological changes. It's coincident with a lot of the earliest signs of cognitive decline. And so there's some hope that if you could fix that problem early on, maybe you could delay the onset and slow the progression of Alzheimer's disease enough that it could have an impact on patients. Okay, so the next question then is, if this is so important, like it's such a big blood flow decrease, it's like, why hasn't somebody fit it out? Is that the vascular network in the brain is quite complicated. So this is the cross-section of the cortex, just the very outer layer of the brain from a rat, where blood vessels have been filled with a dark ink. And basically the problem in blood flow could be occurring at any level of this vasculature. So it could be in the arteries up on the brain surface, could be in these arterioles that plunge down into the brain, could be in the capillary network that they feed, all this like spaghetti-like shape of very tiny blood vessels. It could be in these venules that drain blood out of the cortex, or it could be in the veins that drain blood out of the brain. And so what's needed is an ability to analyze blood flow and see where things are going wrong across all of these different levels of the vascular hierarchy.
So I'm going to tell you a little bit about the tools that have been developed actually here at Cornell about 25, 30 years ago. So the main technique that we've been using to study this in mice is called multiphoton microscopy or two-photon microscopy. And it's a laser-based microscopy that lets us look inside the living brain. So here's some images to give you a sense of how this works. So this is a vial of fluorescent dye in the middle, this green thing here. And what you can see is, actually I'm going to use the mic here. Okay. So what you can see here is that there's a high intensity laser coming in from your right side, the red arrow over here, and it's getting focused into this vial of dye. And if you look very carefully, there's one little spot of green fluorescence that's coming from where the laser is focused in. Okay, so if you use, so that's a special high intensity laser that we call a femtosecond laser, and it is called that because it reaches extremely high intensities in little short bursts of light. So it's a pulse laser rather than something like this laser pointer that I have in my hand. You get extremely high densities of photons coming out, and then a period of time, and then another packet of photons coming out. Okay, on the left side here, we have something that is more like an ordinary laser. And what you can see is then when we focus it into a vial of dye, you get fluorescence from all over. So you get sort of this bow tie shape of light emerging here. And what that means is that if we tried to shine this into the brain of a mouse, we would just see a glow from all over. So the way our microscopy works is we take one of the high power lasers that's pictured on the right. It excites a small volume of fluorescence in just one little spot. And then we take that laser and scan it around inside the sample, and then point by point map out where there is fluorescence inside a sample. And we can do that quite deep into something like the living brain, because we know that all of the light is coming just from one spot rather than a big cone or a big volume. So the other thing that's important for our imaging here is that although this imaging technique can go quite deep inside a sample like the living brain, we can't quite make it through skin and bone. Okay. So what we do is, in these genetically engineered mice that have the genes for Alzheimer's disease, is we actually put a window in the skull. And so what you see here is a mouse that's anesthetized, and at the top of on top of its skull, we've taken a dental drill, the same sort of dental drill that you suffer through in the dentist's office, and we've drilled off a little piece of the bone and replaced that part of the skull with a piece of glass. And then there's basically glue masked up all around the side here that is covering the edges of the piece of glass. So we've literally created a window into the skull of the animal, and what you're seeing inside the window on the right in here is actually the brain of the animal. And so the dark that you can see, those are all the blood vessels. Those are the larger blood vessels that feed the brain of the mouse. So next, though, to be able to look at the smaller blood vessels, what we've done is basically do an angiogram. And some of you may have experienced this. There's actually a clinical procedure that is very similar to this. So to highlight the blood flow, we've done an intravenous injection of a dye, and it goes into the liquid part of the blood, and it flows. And then when we use our microscopy to basically scan around inside the mouse's brain, we can reconstruct some volumes and basically reconstruct movies of what's actually happening. So we see here, this is a movie that is going from the top of the mouse's brain and looping around. So it's just about to loop around. So the top of the mouse's brain, you can see some of those big blood vessels that you could just barely see by eye when you looked at the mouse's brain just through the window. And then this movie steps down deeper and deeper, and you see these sort of like spaghetti-like blood vessels that are getting smaller and smaller. These are the smallest blood vessels that are the capillaries. And they're lighting up because the dye has been injected into the blood vessels and is actually circulating around. If you look very carefully, especially at the smallest vessels, you'll see that a lot of them have sort of a flickering appearance. Some of them have sort of a black and white banded appearance. What those are the red blood cells. So we're at magnifications where you can actually see individual single cells. The red blood cells are showing up because when we do the IV injection, the dye goes into the liquid portion of the blood, but it doesn't get incorporated into the red blood cells. So what we're actually seeing is the shadow, if you will, of each one of these red blood cells. And we have enough magnification that if you do look very carefully, you can see the motion of these individual red blood cells and see that they're actually running through in single file some of the very smallest capillaries.
Great. So now we've got a mouse that gets Alzheimer's disease, and we have an imaging technique, two-photon excited fluorescence microscopy, that lets us look into the brain of the mouse. We can see where blood vessels are, down to the finest vessels, those individual capillaries. And we can even measure blood flow by tracking the motion of those unlabeled red blood cells that are moving around inside the vasculature. Now, just one quick note before I go on. What we're going to do now is we're going to go through kind of a lightning round of lots of data that helps you see what causes these blood flow reductions in Alzheimer's disease mice. But Amy told me there was going to be a quiz at the end of things today. And I'm just going to give you a hint. Two-photon excited fluorescence microscopy is definitely the answer to one question. Actually, that's a technique we should be proud of here at Cornell, because it was invented here at Cornell University in the early 1990s in Watt Webb's lab in the Applied and Engineering Physics department. And it is now the technique of choice globally for this kind of high-resolution imaging inside animals to study disease and normal state physiology. And one of Cornell's most valuable patents. So anyway, this is kind of putting all of this together. So now we're imaging the blood vessels, here shown in red. and those amyloid plaques, shown here in green with another fluorescent dye, in the brain of an Alzheimer's disease mouse. And so we basically just went in and asked an open question, like, where is blood flow messed up in these Alzheimer's disease animals? When we looked at the larger blood vessels on the brain surface, these venules or these arterioles, we didn't see any signs of occlusion or clotting or constriction or something like that that was different in the Alzheimer's disease mice as compared to the non-Alzheimer's disease mice. But when we looked at the very smallest blood vessels in the brain, individual capillaries, we did notice something different. So in those smallest blood vessels, blood cells flow single file. And if you look at this little capillary right here, you can see these little dark spots that are made by red blood cells. And if you look, you can see this is that same capillary imaged over time, and you can see that those dark spots are moving frame to frame. So that vessel is flowing. This one, on the other hand, you can see there's this dark object that's just stuck right there in one spot of that capillary, and it doesn't move. So we called these stalled capillaries. And it turns out that, so this is the fraction of capillaries that have stalled blood flow as a function of the age of the animal for Alzheimer's disease mice in blue and non-Alzheimer's disease or wild-type mice in red. And what you can see is that the number of these capillaries that are non-flowing is elevated throughout the age of the animals. Now, if you look at the vertical axis here, you'll see it's only 2% of capillaries are blocked. And I know what you're thinking. You're like, Chris, you just wasted my lunch hour. Like, if 98% of my capillaries are still flowing, I should be good, right? But remember, if 2% are blocked, that means 4% have lost their source of blood flow because they were downstream from it. And so they're going to be flowing extremely slowly. Another 8% are going to be impacted somewhat. Another 16% maybe a little bit. And almost 10 years ago to the date, when Nozomi and I sort of, in our lab, figured out it was about 2% of capillaries looked like they were stalled in Alzheimer's disease, we just did a back-of-the-envelope calculation. Well, how much of a blood flow deficit do we expect based on how much downstream vessels would slow down? And we got 30%, which happens to be the amount of blood flow deficit that these animals have relative to wild types. And that's what launched us on everything you're going to learn about in the next few minutes here. Okay, so it turns out that these stalled capillaries, they're not clumped together. They're distributed pretty uniformly throughout the brain. So here what we've done is we've traced the network of capillaries in a section of the brain of a mouse. Black are the capillaries, yellow are arteries, blue are veins, and red are the capillaries that weren't flowing. They're just kind of uniformly distributed everywhere. You know, when we publish studies about research, one of the things we always do is we say, you know, we sacrificed, 20 mice in order to conduct these studies or something like that. For this, I feel like we need to have a statement in the paper that says we used 3 undergraduates, almost to the point of death, to do this. So we have dozens and dozens of these kinds of things where people manually traced that entire vascular network in order to produce these kinds of plots. And in fact, you're going to hear in a minute from Pietro about efforts to try to involve the broader public in helping us with some aspects of this data analysis. The next question you might have is like, what causes these vessels to not flow? And it could have been many things. It could have been a small clot in the vessel. It could have been a little constriction, like a narrowing. Could be red blood cells that were sticking. But it turned out that most of these stalls were caused by a white blood cell that was just firmly stuck inside that capillary segment. And that kind of makes sense in light of the biology that we understand from Alzheimer's disease. Namely that those aggregates of amyloid beta, they don't just interfere with synaptic transmission and cause problems for neurons, but they increase the kind of inflammation in the brain. And one of the things when you have inflammation in a tissue, like when you get a bruise or you have an infection or something like that, there's proteins that are expressed on the cells that line blood vessels, endothelial cells that line blood vessels. And those proteins that are expressed, when a white blood cell comes along, the white blood cell will bind to it. It'll crawl out of the blood vessel into the tissue and go try to fight the infection or clean up the injury or those kinds of things. In Alzheimer's disease, you get kind of low levels of that inflammation. And it looks like it's causing these white blood cells to adhere and stick. Probably some of them do cross into the brain. We've never seen that happen. but you do find white blood cells that have crawled into the brain in Alzheimer's disease. But they're definitely blocking blood flow in those capillary segments when they're sticking. A few years ago, we got really lucky. So we were doing experiments to try to figure out what kind of white blood cell was sticking in those capillary segments. I can remember this day, it was Tori Muse, an undergrad in the lab, came in and told me that the experiment with neutrophils didn't work. And as you get to be someone that's leading a lab, there's certain kinds of words coming from people in the lab that make you very suspicious. So when anyone says an experiment didn't work, you have to plug down and understand what that means. And she said, well, I injected the antibody that we were going to use to label neutrophils. So the idea was to figure out what kind of white blood cell it is. We would inject molecules that bind to specific kinds of white blood cells, and those molecules would be fluorescent. So we could tell, is it this kind of cell or that kind of cell or the other kind of cell? So we injected an antibody, a small protein that binds to a protein called LY6G, which is on the surface of neutrophils. And Tori said, yeah, I injected the dye, but it didn't label the neutrophils. Instead, just all the stalls went away. And I said, what, they went away? That's awesome, because that gives us a tool to be able to manipulate this phenomenon so we can see what its consequence is. It also tells us it's neutrophils are the cells that are responsible. So anyway, this is that data. This is the number of capillaries that are stalled after we give a drug divided by the number before. So 1 means no change, 2 would mean it doubles, 0 means they all started flowing. So when we inject a control drug, we basically see no change in the number of capillaries that are stalled. But when we inject this antibody against this protein that's on neutrophils, LY6G, we see that about 2/3 of those capillary stalls go away. So this gives us a way to manipulate this capillary stalling phenomenon so we can see what the downstream consequences are. So of course, the first thing we did was try to examine what happens to blood flow. So we basically measured the blood flow speed by tracking the motion of those red blood cells in some of the arteries on the surface of the brain. And we did that before, and then about an hour after, we gave this drug that gets rid of the stalled capillaries. And if you look, you can see that many of these vessels here, one sped up by 43%, 24%, 53%, 36%. So it seemed like, in this example, that blood flow sped up quite a bit. If we look at this data on average, so this is now looking at the blood flow after the treatment divided by blood flow before. So again, 1 would mean no change. 0 would mean it stopped. 2 would mean the blood flow speed doubled. So when we take a non-Alzheimer's disease animal and we give them the real drug, there's no change in blood flow speed because they don't have stalled capillaries. When we take an Alzheimer's disease animal and we give them a control drug, there's no change in blood flow speed because that doesn't affect the stalls. When we take an Alzheimer's disease animal and give them the real drug, we see about a 30% increase in blood flow speed. And that was true in both young Alzheimer mice as well as in aged Alzheimer's disease mice. So this data basically suggests that this plugging of capillaries, at least in these mice, is what's responsible for the decrease in brain blood flow that they see. Now, of course, the next question is, how does this blood flow increase? Does it improve cognitive function at all?
I mean, I'm sure we all agree that probably having blood flow to the brain is a good thing, but what we really care about is memory. Do we still have the ability to remember, make new memories? Do we have those cognitive abilities? So the next step was trying to test that. We're using mice again. It's a little bit hard to do an interview with a mouse and try to tell if it has these cognitive symptoms. So in the clinic, what physicians will do is they'll give the patient or the putative patient, they'll give them a test that asks questions about memory and different kinds of cognition. But it's a little bit hard to do that for a mouse. So what we do is we actually take advantage of the mouse's natural curiosity and test its working memory or its short-term memory by letting it basically play with toys. So this is the way we try to figure out whether the working memory of a mouse is working. So we put a mouse in a box and we give it 2 toys. They're usually like little plastic figurines, like little Smurfs or little toys. And we let it explore and investigate these toys for a little while. Mice are very curious, so they will actually go and sniff at each toy, and they have whiskers, and they explore with their whiskers. So they will use their whiskers to sort of feel around on each of the toys. So we give it a while to explore both of them. And get to know them. And then what we do is we remove the mouse from the box and then give it sort of a delay of 1.5 hours or so. What we do during that time is we take one of the toys and move it to a new position, but change nothing else about that. So then if we put in a normal mouse, a wild type mouse, back into the box, the normal mouse will remember something about its previous experience, and it will realize that one of the toys is in a new location. So what it will do is it'll spend a little bit more of its time at the toy that moved and went to the new location versus the old toy. However, if you put in a mouse that has the Alzheimer's disease genes, it'll split its time exactly at both of its toys, presumably because it doesn't seem to remember about the experience that it just has, and so it doesn't realize that one of those toys is now in a new position. So this actually gives us a really nice test because it's actually convenient to measure these differences in time. So the way we do that is we use video tracking, and we put a camera that looks down on these cages, and then we basically use the video monitoring to track the position of the mouse's head. And so these are examples of traces that we've taken, where the blue is the location of the toys, and the green shows where the mouse's head has been in the entire recording session. And so we score the mouse as investigating the toy when it's within a certain distance of each toy. So with this, we can very closely track the amount of time that a mouse spends, and so that we can quantify that. And this bar graph here is showing us the amount of time that it spends at the moved toy, the toy in the new position. And so a normal wild type mouse will spend 60% of its time, just a little bit more, at the toy that has been moved. An Alzheimer's mouse spends exactly half of its time at both toys, splitting it evenly because it doesn't remember. Okay, so now we have a drug, though, that we think improves blood flow. So what we did was we tested these Alzheimer's mice, and then we gave it the drug, and then tested it again. So the AD with Alzheimer's, or the AD with the drug LY6G, is now in this gray bar. And very excitingly, it's almost up at the same level as the wild-type normal mice. So this suggests that giving this drug actually improved the memory, so that these Alzheimer's mice seem to recognize that the toy has moved to a new location and act very much like the normal mice in terms of their memory. So the other thing that's very exciting about this data was that we can do this test quite quickly. So we can test the mouse, wait just 2 or 3 hours or 3 hours or so, and then test it again. So these were actually the same mice that we tested first, and then gave a little break of 3 hours when they had, so they had their impaired memory, gave it the drug 3 hours later, and then tested again, and then it was the same mice that suddenly had improved memory. Of course, we have to be careful that they're not like learning better. So we also did an experiment where we took the mouse, ran it through the same sequence, gave it an injection that was not the drug, that's just the vehicle, waited 3 hours, and then did it again. And then here we see no change in the ability of the mouse to realize that it has a newly positioned toy. So this is very, very exciting because this is probably one of the fastest treatments that we've ever seen for improving memory. And while I won't go into all the details of it, it does turn out that this improvement, if we keep giving the drug, we can still get this improvement fairly late into the disease.
Great. So just to summarize, I hope we've convinced you today that about 2% of capillaries are blocked in mouse models of Alzheimer's disease. This is caused by white blood cells that are stuck in there. When we give a drug that inhibits the adhesion of those white blood cells, those stalled vessels start flowing again. And associated with that is about a 30% increase in brain blood flow. And most excitingly, associated with that improvement in blood flow, there's an improvement in performance on these kind of spatial and working memory tasks within a few hours. This is one of the first compounds anyone's described that can rescue the cognitive function of an Alzheimer's disease mouse that already has these kinds of impaired memory performance. And obviously, there's opportunities here to try to develop, to try to push this forward into developing therapies that could be used in humans. But one of the things that really slows that down turns out to be a detail of the analysis of the data that we take. And before I turn it over to Pietro to talk about that, and hopefully to recruit all of you to be participants in helping us analyze this data, I just wanted to conclude by thanking the wonderful research lab that Nozomi and I run. We currently have scientists from 6 continents in our research group. It's a really international, very diverse, and very intellectually vibrant group that's responsible for all of the stuff that I've told you about. And these studies were not free. It was actually quite expensive, and this was funded primarily by the National Institutes of Health, but also by the European Research Council and a number of foundations, including the Bright Focus Foundation. So with that, I wanted to introduce Pietro Venucci, who is the director of the Human Computation Institute and someone who's been critical in helping us overcome what has turned out to be the dominant bottleneck in this research, which is some data analysis steps.
Sorry.
Hello. So, so again, my name is Pietro Venucci. I direct a small research institute called Human Computation Institute. And so you're not going to hear me say words like neutrophil because I'm not a biomedical scientist. I'm a cognitive scientist. And even though I was forced to take some neuroscience courses, what I really pay attention to is cognitive function, sort of the behavioral characteristics of cognition. And in particular, I'm interested in designing collective intelligence systems. So partnerships between humans and machines, or sometimes many humans and many machines all working together towards some goal. And so my institute has a mission of choosing goals that have a beneficial and large societal impact. So that's what I do, and I was very fortunate, of course, to meet Chris and Nozomi because they were working on exactly the kind of problem that we want to help solve. And when I learned about the very time-consuming and painstaking manual analysis of the data that they do and how that was sort of slowing down their research, I realized there was an opportunity there to apply some of these techniques of collective intelligence and collective thinking to help them do the analysis faster. And I do want to say that I love the story about Tori and this serendipitous discovery where she thought the experiment failed, and just call attention to the fact that it takes a scientist with the right kind of background and right intuition to consider a case like that and realize that anomaly is actually important and meaningful, and then take that into a new research track. So if Pasteur had seen the fuzz in the Petri dish, and thought, oh, another failed experiment, and tossed that away, we wouldn't have penicillin today. So it's really to the credit of Chris and Nozomi that serendipitous finding actually turned into this, and it's an honor to work with them. And also just to say, I spent 10 years at a federal research agency looking through lots of research proposals, and I got pretty good at sorting the wheat from the chaff. And so in my first meeting with Chris, I realized, wow, there's something really substantive here, and I'm ready to spend, the foreseeable future in trying to help them be more successful. So, I asked, Chris sort of explained the work they had done and much of what you've seen here, although some of that hadn't even happened yet. And then, at a certain part, I asked to see, well, what is this, this analysis that's taking so long to do? What does that look like? If I'm the laboratory technician, what is the task I'm doing? And so I guess one of these is going to make something light up. He said, well, the first part is we have to sort of trace this extensive vascular network. We have these 3D images that we get from the mice, and we're analyzing the blood flow, and we get these networks, but in order to see what's happening downstream, we actually need to model these computationally. And then we have to go through and find out which of all these individual vessels are stalled in the way that he described before, and then map these back into this network. Well, wow, that is a lot of work, and I can see why it would take so long. But I got extremely excited very quickly because I looked at both of these pictures and they were both instantly familiar to me because I was aware of other projects in this space of collective intelligence, also called citizen science projects, where members of the general public participate often in an online kind of game-like environment to help analyze data. And so that gave me great hope that we would be able to do something along the same lines with their data. So the one that I resonated sort of the most quickly with, so over on the left side here, you can see this is what Chris and Nozomi were showing you earlier, where you have this this blood vessel in the brain of a mouse, and you're trying to decide if blood is flowing or not there. And I saw that picture, and it reminded me of something I did in 2006. I played this online game called Stardust at Home, based on a NASA mission. So they took a satellite, put it in space, flew it through the tail of a comet, and they had these big aerogel collector grids. And they captured some of the dust from the tail of the comet, brought it back to Earth. And then the scientists took a bunch of microscopic images of the aerogel so that they could look at each image and try to find these little dust particles, because it would be the first capture of interstellar dust and would help them revise models of how the universe formed in the first place. The problem is they ended up with 1 million, that's not an exaggeration, 1 million digital images. And they did some rough napkin calculations and realized it was going to take them 100 years to analyze the data. So, they created this online platform, and they got 30,000 so-called dusters to volunteer to go through and use this virtual microscope online and look through these pictures of aerogel to find the dust particles. And these 30,000 people over a period of 6 or 7 years found the first 7 interstellar dust particles. So I thought, well, wait a minute, that virtual microscope interface, could be used exactly to do the kind of work that those poor lab techs are doing in the Schaffer Nishimura lab. So I contacted the person who created that, and I explained the whole situation, and that's Andrew Westfall at Berkeley. And he said, I get contacted a lot about ideas like this. He said, but and most of the time it's not really a good fit. And I have to say no. He said, but you're right, this is a really good fit. And he said, and by the way, I had a personal loss to Alzheimer's, and I really want to help you make this work. And he personally, and he and his team worked together to help us develop our prototype system, which involved literally taking their Alzheimer's data and putting it in Stardust at Home. And it worked out of the box. So we knew we were onto something good. There was another citizen science platform called iWire that looks at retinal neurons in the brain and does this kind of mapping. So we were going to go to them. In fact, they were part of our team at the beginning, and we were looking for a plan to build a platform based on iWire to do this kind of mapping of the vascular network. It turned out, though, that so they have a student in their lab named Mohammed Haft Jafaharian, who was very interested in machine learning and artificial intelligence. And he went and spent time with the iWire scientists, and he came back with all that knowledge, and then he created an automated system that could do that, which is even better, because we're not going to waste human time if we can do it with a machine. So now, today, It's his algorithm that does this part for us and figures out where to draw those outlines. And then we went ahead and we, oh yeah, this is the example. This is where you can see we actually put the Alzheimer's data into Stardust at Home, and we just changed the name. All done, right? But we've made some improvements, and we've gamified it. We've made it more, hopefully, kind of engaging for participants and adapted to this particular task. But this is what this game called Stall Catchers looks like today. And anyone can go to stallcatchers.com and you know, go through this brief tutorial. It walks you through how to play the game, and then you're actually helping to analyze the data. So whereas previously, you know, it would take 6 months to a year to analyze a single data set, now with you know, many people playing stall catchers, we can do it faster. So one of the issues that comes up right away is if you have members of the general public analyzing the data, what guarantees do you have about the quality of the analysis? How do we trust random people, you know, to come up with the right answer? Well, in this collective intelligence sphere where I do my work, we have methods of combining answers from many different people about the same vessel to come up with a single expert-like answer. So one of the first questions we ask is, if we're going to just take a simple average, let's say that every single person, I show you a vessel, every single person in this room says flowing or stalled. How many people's answers do we need to combine before we feel a certain level of guarantee about the quality of that answer? And we have very, you know, specific kind of requirements on the research side about how good that answer needs to be, how often. And in our initial test, we found that with, you know, five people wasn't enough, 10 people wasn't enough, 15 wasn't enough, but somewhere between 15 and 20, we were able to sort of cross this threshold on both sensitivity and specificity, two ways we measured the goodness of the data quality. And so we basically validated the approach. Not only did it work in the Stardust interface, but we could actually produce, with enough people participating, the lab quality data. Well, then the problem becomes, how do you get lots and lots and lots of people to do this? Because now if you need 20 people for every single lab tech, you're going to need a lot of people to do this. So we did validate the approach, but so then part of my work as a scientist is to come up with better collective intelligence methods. And so I did some theoretical work, and we were able to combine answers in a more sensible way. Instead of just taking the average of everyone's answer, we started sort of tracking people's performance in the game and realizing that, well, this person is actually really good at distinguishing between flowing and stalled, so we should assign a little more weight to that person's answer. This person's new, they just started yesterday, they're doing a little better than chance, but not much, so we'll assign less weight to that. And because we know sort of how skilled each person is, we'll continue to gather evidence only until we have enough to have an expert-like answer. And so by combining these methods in the right ways, we were able to reduce our magic number to 7. So now it only takes 7 people on average to come up with that expert-like answer. And we think that's actually pretty close to the theoretical limit of what we can do. And of course, one thing that helps is to continue to educate our volunteers and give them opportunities for learning, because the more skilled they become, the smaller that number gets. So we have over 15,000 people registered on stall catchers today. So that doesn't mean we have 15,000 people playing every single day. It ends up being probably 100 or 150 people, and it's usually 1% of the registered number. That's typical in these online systems that are playing on any given day. But combining answers in this way, we're analyzing data 5 times faster than in the lab. That's already an improvement from several months ago when we were 3 times faster. So I had to update the slide, which was very satisfying in this respect. And what does that mean in practice? It means that if it used to take 6 months to answer a research question, we can now do it in only 1 month by analyzing the data. I won't say too much about this, but we're doing some sort of interesting research in different ways of doing human and artificial intelligence partnerships. So as I mentioned, Muhammad came up with this algorithm that can automatically now trace the vasculature, which allows us to draw the outlines. And that tells the person who's playing the game, which vessel should I be paying attention to? And so the partnership here is that the machine draws the outline and the person says what's happening inside of it. So that's a nice and effective partnership. We're also, we've had our players tell us when the outline is going around something that really doesn't look like a vessel at all. In this case, it's crossing over a very large vessel, which is not a capillary. And they flag that for us. So we collect all these flagged examples of bad movies, and then we use those examples to teach a machine to remove the bad movies ahead of time, and then people don't have to waste their time. So again, we're trying to save players time by pulling out those bad movies. And then finally, there are some vessels that are easy enough to tell the difference between flowing and stalled that we can teach machines to do those. We just have to make sure the machines do a good enough job. So today we have a system that we're still testing, but it looks like it can confidently analyze 10% of the vessels just the way the humans are doing it. That's 10% less that the humans have to do. So the last thing I want to tell you about is that we often run events. This is a way we get people to know about our project. People who are already involved in the project love our events because it's kind of it's an exciting competition, and there's often just a lot more people playing, and the leaderboards get active, and we have a live chat box. There's more chatter. But this is at a level we've never done before. And the only way we can do this is because we're partnering with a lot of different organizations to make this possible, including the public libraries. So we've done international events on all 6 continents. This is more of a national event we're calling the Megathon, and it's 2 orders of magnitude at least bigger than anything we've done before. Our goal is to get 100,000 people playing the game all at the same time for the same 1-hour period of time on April 13th. We're going to put an entire data set that would normally take, again, 6 months to a year to answer in the lab. We're going to collect 1 million annotations in that 1 hour. And in the end of that 1 hour, we're going to answer a research question from 1 year to 1 hour. And we're going to break a world record. So I would invite you to join us. And so the next thing I'd like to do, if I don't break anything here, is show you this trailer that we created. I hope you enjoy. Thank you.
Great. So that's all we have today. We'd be happy to take any questions and even stick around afterwards to chat.
Let's suppose that you're writing a really important email to a colleague.
Autoplay.
Or a post on Facebook.
Okay, great. Yes, please.
About those white blood cells that adhere to each other, and if I understood you correctly, they do that because they detect the inflammation.
Correct.
So when you get them to not adhere and increase the flow, the inflammation stays.
Correct.
Will it get worse, and does it have negative effects, other negative effects?
Yeah, those are all great questions. So the question is, so if we block the adherence of white blood cells, we're not treating the thing that caused the white blood cells to stick, the remaining inflammation. And yes, I agree that underlying inflammation could be causing all kinds of other problems. In fact, it's known that it interferes with the ability of brain blood vessels to dilate in order to adjust blood flow to the brain when one area is more active or things like that. And so it's true, we would not be treating, if all we did was block white blood cell adhesion, we wouldn't be treating the underlying cause. There's another reason that directly blocking white blood cell adhesion isn't a great idea, and that's that it would basically immunocompromise you. So if you block white blood cell adhesion in one spot, you're going to block it everywhere, and that's going to defeat the ability of your body to fight off infections, repair wounds, those kinds of things. So while directly blocking white blood cell adhesion has been a great tool for us to understand how these stalled capillaries impact blood flow and they impact cognition, I think it's somewhat unlikely, although Nozomi and I disagree about this, we'll talk about that. I think it's somewhat unlikely that that becomes the therapeutic approach, if indeed the same thing happens in humans. So we have a parallel research program where we're chasing what is happening in the endothelial cells in the brain to try to identify something that is brain and Alzheimer's disease specific. And the idea would be to block there, to directly shut down the vascular inflammation and not compromise the ability of white blood cells to do their job elsewhere in the body. Now, before we even get to that, though, we have a more fundamental research question, and that's, is this same mechanism at play in humans? So we've shown in mice that this is what causes decreased brain blood flow, but we don't know yet that that's in humans. And it could be that directly blocking white blood cell adhesion is the way we answer that question. So there's a bunch of drugs that have already been developed and have passed phase 1 trials in humans or are even on the market, that the way they work is by blocking the activation, the trafficking, the binding, various states of kind of that inflammatory cascade. They're mostly given for autoimmune disease, like rheumatoid arthritis or MS, things like that. They haven't been tried in neurodegenerative Alzheimer's disease because there was no reason to. But they're safe, at least for a short time. So we're currently screening some of those drugs in mice, ones that, based on how they work, we think they're likely to interfere with this leukocyte adhesion. If we find the one that works, we would then go do a short-term trial in humans, where we just assessed, if we give this drug, does the blood flow to your brain increase? And if the blood flow to the brain increases, is there any improvement in cognitive function? If that turned out to be true, that would really put the wind behind the sails for a massive, sort of target finding and drug development effort that looked to intervene in a place that wouldn't cause those immunocompromising symptoms. So in terms of like how we see this moving forward, that's kind of the roadmap we have right now. But chasing those upstream molecular mechanisms, each one of those is one of those year-long research questions for us. And that's why stall catchers is so critically important, because it dramatically accelerates how quickly we can answer those questions.
Who are your collaborators elsewhere outside of Cornell?
Yeah, we actually have. So in addition to the stall catchers team, there's quite a few collaborators, and some of the critical ones are at Weill Cornell Medical School. So we work a lot with Constantine Iodacola. But we also have a network of people outside of that we work on various parts of these different questions. So it is a group effort here. And some of those collaborators are on the technology side. Some of those collaborators, for example, are on the computational analysis side as well. So it's, if we put up the names on the board, it is dozens of people. So if we actually just recently published a paper about this, and one of the things that I like to point out is not just the senior collaborators that are on there, it's quite a few students and undergraduates at Cornell that were essential to this as well.
Yeah, so here's the paper that's published. This represents authors from 5 different laboratories, France, Spain, Rockefeller, Weill Cornell. Please.
People vary as to how dizzy they get when they stand up quickly. Is there any possible correlation between that susceptibility and susceptibility to Alzheimer's?
So the question is whether that sort of feeling you get when you feel dizzy when you stand up, whether that could be viewed as like a risk factor or something like that for Alzheimer's disease. I don't think so. Don't know for sure. But how long you feel dizzy when you first stand up has a lot to do with how quickly this thing called a baroreflex where you get this dilation of blood vessels in the neck and the brain in response to decreased blood pressure. It sort of depends on how quickly that activates. I don't think if you feel dizzy, like if you tend to feel dizzy when you stand up, that's somehow a symptom of some deeper underlying problem. Yeah, I wouldn't sweat it. Just make sure you don't fall back over. Yeah, please.
Professor Schaffer, so when you are collecting data on mice, they are quite social animals. Do you also, by the way, you're doing an experiment anyway, collect data about their spouses or their parents, caregivers? Do they have a more correlation, like a spouse or caregiver or parents or children are more likely to get Alzheimer's disease than the general public?
Yeah, that's a really interesting question. So our mice, they are housed in sort of an artificial condition here. So they are in cages, so they don't have quite necessarily the full social aspect that you would expect in naturally born mice or mice that are living in a real environment or something that mimics the human. However, one of the things that we do know from mouse studies is that, like in humans, the social interactions do matter for the progression of Alzheimer's disease. And so one of the variables that we are starting to keep track of is how they're caged. So it does turn out that if a mouse is by themselves in a cage for their the entire time of the experiment, that they seem to actually pick up some of the symptoms of depression, and that does actually affect how Alzheimer's disease progresses. So one of the reasons that we have to do these studies in models like Alzheimer's disease rather than like a reduced model, like a cell culture model or some of these engineered things, is that a lot of these different variables, like the immune system, like some of these things like activity, depression, stimulus, all of these things actually do accumulate and matter a little bit. So it is a very interesting question and something that I think quite a few people are interested in, how all of these systems interact.
Yeah, absolutely. But in terms of like which mice do and don't get Alzheimer's disease, that's governed exclusively by which ones inherit the genes that are inserted into their genome. But definitely the social experience of the mice throughout life can alter how that disease develops in the ones that carry the mutated genes. There was one more? Yeah, sure.
Is there a reason why it's happening in capillaries and not in arteries?
Yeah, so, well, that's a very deeply profound question. So the question is, why is this happening in capillaries as opposed to other blood vessels? So I don't know the answer. I'm happy to speculate, but this is sort of like, first you walk out on a limb and then you're on a little twig and then you're like balanced on the leaf. That's sort of where I'll be. So the way I think about it is if you have large scale inflammation, like you've had an injury or a bacterial infection, there's a massive upregulation of these proteins along the blood vessel surface. And then you get white blood cells that stick to all different kinds of vessels and crawl from those vessels into the tissue. When people studied this, the easiest vessels to see were kind of arterioles and venules, and veins. And what people noticed is that white blood cells stuck in the veins and crawled out into the tissue from the veins. And that became this sort of dominant understanding of where inflammation happens in the body. That when you get inflammation, it's white blood cells adhering in veins, crawling into the tissue and doing their job. And people didn't really have tools to be able to look at what was happening in the capillary beds. And there was all this exciting biology happening in the veins. So for 20 or 30 years, that's where kind of inflammation biology is focused. My guess is when you have lower levels of inflammation, like we have in these sort of chronic Alzheimer's disease, it's just not enough for these white blood cells to stick and hold on to the vessel wall in an artery or in a venule. But in a capillary, the cell goes single file. The white blood cells actually have to distend and distort a little bit to squeeze through a capillary. So they're touching the capillary wall all the way around, not just at one little spot. And so even a small amount of inflammation would be enough that cell could stick and arrest there. And probably, like, I think nobody's looked carefully, probably in these cases of more severe inflammation, in addition to seeing the white blood cells that are crawling out of the vessels in the veins, you would see them stuck all over the place in the capillary beds as well. Yeah. Please hear that.
Is there a potential for the stall catcher's way of collecting data to make humans a sort of obsolete part of the process and solely rely on like machine learning algorithms for collecting the data?
That's a great question. I mean, that's our goal.
Yeah, it sounds kind of funny to say we want to make humans obsolete, but in this case we do, and we see this happening in other similar platforms. So in the iWire platform that I described, they've kind of been trending in that direction where the machine part of the work is getting so good that now it's only the very most challenging parts of the problem that are left for the humans. And I think this is a general trend. So, there's a lot of kind of sensationalism around artificial intelligence today, and this concern that it's going to replace all the human jobs, and there will be nothing left for us. And I think what we're seeing is another kind of industrial revolution where the more tedious and repetitive things are now being absorbed by machine learning and AI, and the more creative stuff is left to the human. So a nice example in stall catchers, where a human has an edge over machines, is that a machine is only trained based on the pixels. If it's not in the image itself, the machine doesn't really know it, the way that most people use machine learning and artificial intelligence today. But anyone who's grown up and played with a garden hose knows that if the water goes in one end, it has to come out the other end. So we have this real world knowledge about how basic physics works. And so when you're looking at a vessel and you can't really tell from the image, from the animation, is blood flowing through this or not, but you can see where it might be coming in and where it might be going out, you can infer from that the only way it could be coming out here is if it's flowing through the vessel. The machines don't know how to do that. So I think we're still a ways away from the machines taking over everything for us.
And we tried. I mean, before we partnered with Pietro, we tried to develop a number of algorithms, not machine learning based, but deterministic algorithms to distinguish flowing and stalled vessels. And we could get 75% right or something like that, but that's not even close to the sensitivity and specificity we need to make real progress on these research questions. But I could teach all of you how to score these things as flowing or stalled in 5 minutes. In fact, there's a little video that does it in exactly 5 minutes.
If we are able to maintain the blood flow rate at young age, at old age, do you expect that Alzheimer's disease will not take place?
That's a really great question. So the question is, if you identified someone who was having this decreased brain blood flow early on, and you continuously treated them to try to keep the blood flow at a high level, what would we expect the outcome to be? So I want to be clear, this is not going to cure Alzheimer's disease. If we did that, it's not going to cure it. But to tell you the truth, I think anyone who tells you the goal is to cure Alzheimer's disease is lying to you. It's not our goal. Our goal is to delay the onset and slow the progression so that you can die a better way. Like Alzheimer's, it's the absolute worst way you could go. And so we want to have a different way. So for this, I think would be helpful for that. So for a couple of reasons. One, at least in the mice, we know that if you improve blood flow, even with all of the other pathology still going on in the brain, if you improve blood flow, it's enough to improve cognitive function quite a bit. Additionally, we know, I didn't show the data today, but we also know that if you improve blood flow for a period of time, it improves the clearance of amyloid beta out of the brain. So it looks like as you age, the clearance pathways for amyloid beta out of the brain start to fail. And that's what leads to increased concentration of amyloid beta. It's not that you make more of it when you get older. Now, there's many different clearance pathways, and it's not clear which one is the most important, but definitely clearance through the blood is one of them. And if you have decreased brain blood flow, that's like plugging the drain a little bit, right? And so the amyloid beta can't get out. If the amyloid beta is there, it aggregates. If it aggregates, it drives the disease. So there's a bit of a positive feedback mechanism there between amyloid beta and between these capillary stalls. So we've shown that if we keep blood flow elevated for a month or so, that decreases the concentration of some species of amyloid beta in the brain. So those two pieces of evidence, if the same mechanisms turn out to be true in humans, I would say it gives us hope that if we had a good drug and you could identify people early, that would delay onset and slow progression of Alzheimer's disease. And that's already something that would be a huge benefit. The other thing I'll say is that this kind of symptomatic treatment, right, where we're trying to treat a downstream symptom of the disease, is completely complementary to other kinds of therapies that might be aimed at going after more upstream mechanisms, like trying to interfere with production of amyloid beta or its aggregation or underlying inflammation in the brain. But no, this is definitely treating a downstream effect. It's not something that's targeting the causative aspect of the disease. So I wouldn't characterize it, even if everything went perfectly, as a cure. Great. Thanks so much.